Okay, so now it's noon. So I'm going to welcome everybody to the CABC Center on Addiction and Behavior Change seminar series. And today we have our second speaker in our lineup. We're going every couple of weeks. And then just to look ahead a bit, actually our next event is going to be on the 16th of September, where Laura Slotsky is going to be from Duke is going to talk about leveraging functional selectivity of neurotensin receptor 1 mediated treatment of addiction. And so that'll be in 1 week. And then actually on the 25th, it's not going to be on a Wednesday, but a Friday, we're having our day-long symposium on substance use prevention with equity, a community and neuroscience-based approach. So I've sent out the materials about that, and this is together with the Durham Community Group TRI for Resilient Youth. And it's a Durham outreach group. Wanda Boone, Dr. Wanda Boone is the director of that, and she and her group has been collaborating with DIBS and CABC, and we put together a very informative program with community activists and academic researchers having joint presentations. So I encourage you to do that. And then we go on with our seminar series. Walter Sennett Armstrong will speak on October 7th, and then we go on basically twice a month to the end of the semester. Now, if anybody has a recommendation for next semester, I'm putting together the schedule then. If you would like to present, or if you have anyone actually globally you'd like to bring on, I'll contact them. At this point, we're going to be continuing in the Zoom format, and given that it's pretty low cost to engage out-of-town speakers and even out-of-country speakers, then it actually opens up a lot more doors. So we're hoping to get back to live face-to-face meetings sometime, but probably won't be until next academic year. So anyway, now I'd like to present today's speaker, Dr. Scott Parnell, who's an assistant professor in the cell biology and physiology department at UNC, and is also a member of the Skipper Bowles Center on Alcohol Studies in the Carolina Institute for Developmental Disabilities. And he is going to speak to us about his research and interactions among alcohol, cannabinoids, and primary cilia during early development. Okay, Scott.
All right, let me share my screen. Thank you, Ed, for inviting me. Thank you for having me. All right, everyone sees that okay? All right, good. Okay, so I'm going to talk today about two different stories. I don't usually like to give seminars on two different stories, but today I'm going to talk about two different stories that were begun by people in my lab quite independently. They were working on two completely different projects, but they very fortuitously at the end sort of came together, and they realized that they were both working on what was really a very similar basic cell biological mechanism of how these drugs are interacting. Okay, so I know a lot of you know what fetal alcohol syndrome is, but to make sure that everyone's on the same page, fetal alcohol syndrome was defined a little more than 40 years ago, and its definition is that the child presents with cognitive and behavioral deficits, these pre and postnatal growth delays, and then some very characteristic craniofacial abnormalities. And these include the indistinct philtrums, that little notch above your lip, a thin upper lip, these short palpebral fissures, and those short palpebral fissures result from small eyes or microphthalmia. And this occurs about 0.2 to 0.7% of the population. But that's not the complete story of what alcohol does. Alcohol has a lot of other effects, largely on the brain. So the alcohol can affect the brain at any point in development, even through adolescence. And what's interesting is that not all children present with those craniofacial malformations of FAS. And so we put these children under the umbrella term of fetal alcohol spectrum disorders. So it sort of describes the global effect of prenatal alcohol exposure. But and of course FASD occurs in 2 to 5% of kids. So it's really quite common. It's more common than autism spectrum disorders, etc. But the biggest question that has come around in the field is, why do we get such a big effect? You know, some kids present with these craniofacial malformations. They have pretty low IQs, pretty severe brain effects, and other kids don't. They have more subtle defects. We even just, ADHD in the most mildest of cases. Well, we've learned over the years that there's a number of modifiers of alcohol's effects, and these can be genetics, and I'll talk a little bit about that. Nutrition, we know poor nutrition exacerbates the effects of prenatal alcohol exposure. Other drugs, the dosage of alcohol. And so, the more alcohol you consume, the more you consume in one sitting. Binge drinking, that exacerbates the effects of alcohol. And then, as I'll go into in a bit, the timing of exposure also has a big effect. And so, one of the nice things about these craniofacial malformations with prenatal alcohol exposure is that we can really nicely model these in mice. So here is a normal kid with a normal face, with a normal philtrum. He has nice big eyes. And here's a kid with fetal alcohol syndrome. You see that smooth philtrum, small eyes, and a small head. And here's a normal mouse fetus with a normal mouse lip, normal mouse eyes, normal head. And here's a mouse exposed to alcohol. And you see these small eyes, this smooth philtrum that kind of models what we see here in the kid with FAS, a small head, and these craniofacial malformations are reflected in the brain. So here's a slice through the brain. You can see the anterior commissure here, a bit of the septal region. And here you can see that we've really, this is a pretty severe malformation of the brain, and it's bordering on holoprosencephaly. Sorry, it is holoprosencephalic. And you can see that we've decreased the septal region. The anterior commissure is messed up. Well, we've known for a while that those craniofacial and severe brain malformations resulting from alcohol exposure are due to a gastrulation stage alcohol exposure. So in the mouse, this occurs on day 7, and in the humans, it occurs on day 17. So really very early alcohol exposures. But in the mouse, we can give a single acute exposure to alcohol on day 7 and cause those craniofacial and brain malformations. And if we do that, we get a whole spectrum of effects. So we don't affect the entire litter exactly the same, but we get, so here's a normal mouse face, a normal fetal mouse brain. And here you can see the face is slightly affected, the brain is slightly affected. And as we go down the spectrum, we can go all the way to rather severe effects where the two sides of the cortex are fused together. So that's holoprosencephaly. You can see we're missing an olfactory bulb up here. And you can get some mice that are really severely affected. You can see this mouse has no eyes. It has almost no cortex. And so, that makes sense because the brain and face are developing together and alcohol is affecting both of them. And all of these things we've known for a while that these effects of alcohol exposure during gastrulation are effect of reductions in sonic hedgehog. So for those who don't know what sonic hedgehog is, if we take a section through this embryo, so this is an embryo, it's about day 8, we take a section right through the middle and look at it from the side, and we stain for sonic hedgehog. Sonic hedgehog is expressed on the ventral surface of the embryo, and it's a really important morphogen. It's just quite simply, it's a growth factor. and the embryo absolutely needs this sonic hedgehog. So oh, my apologies. I am that one messed up. Sorry about that. So here's just a section through the neural tube. You can see that it sonic hedgehog is expressed on the ventral surface there, and it is absolutely critical for normal neural tube development. And so we've known for a number of years that alcohol reduces this sonic hedgehog signal. So here's a normal mouse embryo. Again, this is the sagittal section right through the middle of the embryo. Here's that nice normal sonic hedgehog expression. And here in alcohol, it's vastly reduced, especially up here in the head region. And we've known for some time that this loss of sonic hedgehog signaling is due to apoptosis in the developing neural plate. So alcohol kills these cells that are going to form sonic hedgehog, and so they don't make sonic hedgehog and they don't release it out into the rest of the embryo. And so the embryo just doesn't develop because, like I said, sonic hedgehog is an important growth factor. So that led us to raise the question of what happens if we have mutations in sonic hedgehog. We know that if you completely wipe out sonic hedgehog in the embryo, it's lethal. The embryo will die. If you have one copy of sonic hedgehog, in mice especially, there's no effect. The mice, so here's a male mouse with just one copy of sonic hedgehog. He grows, he's fine, he can reproduce. And if we breed him to a female mouse, they'll have the normal Mendelian ratio of half heterozygotes, half wild types. And all of these mice are normal. You could not tell the hets from the wild types. So what happens if we add alcohol? So now we're looking at a gene-environment interaction. And here in our sonic hedgehog wild type litters, we get an incidence of craniofacial malformations, just as we would expect from alcohol, because alcohol is a teratogen. We've known that for a long time. But we can more than double the incidence and the severity of those malformations if we're missing one copy of sonic hedgehog. So in this GLE2, this is downstream of sonic hedgehog, we get that same effect with GLE2. This is a transcription factor downstream of sonic hedgehog. And so we're very nicely showing that we're getting a gene-environment interaction here that where we can greatly exacerbate the effects of alcohol by just having this mutation in one copy of sonic hedgehog. And we see this in with lower levels of alcohol. So we can give levels of alcohol that don't cause long-term behavioral effects. Say here we're looking at an open field, just looking at the time in the center. If we give, there's no effect of the genotype, so knocking out one copy has absolutely no effect. In the wild type offspring, given a low dose of alcohol, we see no effect. They look just like controls. If we give alcohol in these heterozygous mice, suddenly they're spending far more time in the center, which as you know, for a mouse is really abnormal. Mice don't like to spend time in the center of that open field. They want to spend it around the edges where they're safe from predators. And so this is a really abnormal disinhibition, but it also further goes on to show us this gene-environment interaction that we see. And so a number of other studies have kind of led us to building up this pathway. I'm not going to go into this whole pathway, but we've kind of built up this whole pathway where we have this interaction with growth factors, which, as I'll show in a minute, are present on these primary cilia. And this interacts with other cellular machinery to either go through normal cell proliferation, which you have to have in a developing embryo. You need a lot of cell proliferation, versus the control of DNA damage. When you have lots of proliferation, you have the potential for DNA damage. So there's a lot of cellular machinery to kill off cells that have too much DNA damage. And so there's always the potential for these cells to undergo apoptosis. And that's And like I said earlier, we know that alcohol causes apoptosis. And so we asked the next question, well, what if we knock out BAX? So BAX is this protein here. It's a pro-apoptotic mitochondrial protein. And not in normal cells, it's quite quiescent. It doesn't really do much. But under cellular stresses, it induces apoptosis. So what if we take out BAX? And BAX, you cannot take out both copies of BAX, and the embryos are still mostly normal. And if we take out, and if we give alcohol to our wild types, we get a percentage of eye defects. And so a lot of what I'll show are eye defects, because eye defects are really nice to see. So here's a mouse fetus with no eyes. We can look at these eyes and we can score them. We can rate how many are abnormal, etc. And if we take out one copy of BAX, we don't really get much of an effect. But if we take out both copies of BAX, we can almost completely protect against the effects of alcohol. It's really quite startling how effective this is. And so this just shows that we can go both ways. We can knock out sonic hedgehog and exacerbate the effects of alcohol. We can knock out BAX and protect against the effects of alcohol. So that there are very significant gene-environment interactions here. So this was all with the gastrulation stage exposure. It's this gastrulation stage exposure where you get these really severe craniofacial malformations. But what happens during neurulations? In the mouse, neurulation precedes gastrulation, so it happens just a couple of days after. And it's, neurulation, for those who don't know, it's just simply the process of taking that flat neural plate and forming a neural tube. And so during neurulation, which is, we're going to look just a couple of days later. So here's gastrulation in the mouse. A couple of days later, we've gone from this flat neural plate into, we're starting to form a nice round neural tube. So alcohol during this exposure actually doesn't cause any of the craniofacial malformations, with the exception of eye defects. But we don't get the effects on the lips. We don't get all of the other severe brain effects. But we do get brain effects. We still get actually less severe, but we still get very similar brain effects. And you can't see here, but we get, if you look at the underside of the brain, we actually get some similar but less severe effects of alcohol. And what's interesting here is that even though we get effects on the eyes and on the midline parts of the brains, which you can kind of see here if you compare this one to this one, we're really affecting some of these midline structures of the brain. But what's interesting here is that unlike a gastrulation stage exposure, with a neurulation stage exposure, we get effects of alcohol, but they're not through an apoptotic mechanism. There's in these parts of the neural tube that are going to give rise to these parts in the brain, there's just no apoptosis. Alcohol is working through some other mechanism besides apoptosis. So what is this mechanism? So this really puzzled me for a long time, because this was started as some work I did as a postdoc, and it kind of confused us how alcohol so very much caused apoptosis during gastrulation, but just a day or two later, there was no apoptosis. So we just sort of wrestled with this for a while, until we started thinking about how sonic hedgehog signals. So almost all cells have these little appendages called primary cilia. And these primary cilia act as antennae. So they're sitting out here in the extracellular matrix. And what their job is essentially to transduce signals from outside the cell into the cell. So they take, there's lots of receptors on the cell membrane out here, and they can take these signals and very nicely transduce them into the cell. And so we'll take sonic hedgehog, for example. So when sonic hedgehog is not present, patched one is the receptor for sonic hedgehog. Patch 1 inhibits smoothing. Smoothing is a G protein coupled receptor. And so smoothing just kind of hangs out here outside of the primary cilium. And under normal circumstances with no sonic hedgehog, you have, through a whole series of mechanisms, you have a high pKa levels here in the cell, okay? And these, not in the cell, just in this primary cilium. And these pKa levels cause the GLI proteins, those transcription factors I mentioned, the end product of the sonic hedgehog pathway. These GLI proteins go from a repressor to an activator form, and they're cleaved very intentionally within this primary cilium. And when sonic hedgehog is not present, their activation is, these GLI proteins are not activated, so they don't go out and induce transcription and normal cell proliferation. When you have sonic hedgehog, it binds to patched, it releases its inhibition of smoothing, this G protein coupled receptor. Smoothing can then translocate to the primary cilium, where it's coupled with its inhibitory G protein. This G protein inhibits adenylyl cyclase, which then decreases pKa levels within the primary cilium, thus activating these GLI proteins. And these primary cilia are fantastic little organelles because they can really concentrate pKa levels, and they can very quickly and nicely adjust pKa levels because they are their own little compartment. So you don't have to adjust pKa levels in the entire cytoplasm. You just do it within this primary cilium, and then you can have a big downstream effect. And when sonic hedgehog is present, you undergo nice normal cell proliferation. And so the first question we asked was, well, what if alcohol is affecting these primary cilia in some way? You know, we know that we're reducing sonic hedgehog in some, or the downstream products of sonic hedgehog. What, maybe we're affecting these primary cilia in some way, because we need to understand how alcohol is working through a non-apoptotic mechanism. And so the first thing we did, and this was done by Karen Boshin, a postdoc in my lab, is she looked in the neural tube and she looked for the number of primary cilia. So primary cilia very nicely stained with this ARL13B. It's a small GTPase that's present in the primary cilium. And so here are these cells of the neural tube. So here's the neural tube. We're looking at just this part of the neural tube right now. And here's these primary cilia. Here's a close-up of these primary cilia. And long story short, we don't find a difference in the total number of cilia within the neural tube. Alcohol doesn't seem to be decreasing the number of primary cilia. So what about function? You know, these primary cilia have a lot of important functions during development, and in fact, there's a whole section of developmental biology that is looking at these functions of primary cilia. And when you have defects, genetic defects in these primary cilia, you get what is known as a ciliopathy. And so there are a number of ciliopathies that are developmental disabilities, and they can cause quite severe developmental defects in kids with these ciliopathies. And so we looked at one of these GLI transcription factors, GLI3, and we find that even though we don't see differences in the total number of primary cilia in the neural tube, we're getting a significant reduction of the activated GLI forms. So so alcohol is reducing the downstream processing of these GLI proteins. And when you look at the cell cycle gene expression that is downstream of GLI proteins, you can see that they're significantly reduced in. So alcohol is reducing some of the effects, or some of the cell cycle gene expression, and reducing cellular transcription in the neural tube. And to go back to some of our gene environment studies, we wanted to go back and see if there was a gene environment interact. So KIF3A is a really important protein that in the primary cilia. Actually, if you knock out KIF3A, both you get a ciliopathy. So it is a pretty important ciliopathy gene. We took out just one copy because we didn't want it to be lethal. But when we do, we see that we get a pretty severe effect of alcohol that we don't see in the wild types. So we can exacerbate it with this. So again, we're seeing this cilia gene and ethanol interaction here. And again, I won't go through this too much, but we also see this effect on various behaviors. Again, we're looking at center time in this open field test. You know, we get a small effect, non-significant effect in our wild type GIFs in open time, or the center time. And this is exacerbated in our heterozygous mice. So we're getting a gene environment interaction, both looking at fetal brains as well as long-term behavior. And so some subsequent work that we just published, we're looking at RNA sequencing. You know, we're starting to build this whole story of exactly how alcohol is affecting these primary cilia. And for somehow we're still not quite sure, we're still working on, but alcohol is affecting a number of these primary cilia genes that regulate how these cilia function. And so alcohol is down-regulating some of these genes and just making them not able to work like they're supposed to as well. And then, you know, we still have a long ways to go with this story of figuring out exactly how alcohol is affecting these primary cilia. But we now know that alcohol is affecting development by affecting these primary cilia. And so I think the important things to conclude here are that, you know, we get these gene environment interactions. They're different based on when the embryo is exposed. So we get different effects of alcohol during gastrulation versus neurulation, for example. And so during gastrulation, alcohol seems to have this apoptotic mechanism effect, whereas during neurulation, just one day later, we tend to get this primary cilia effect. And so what we have proposed is that even though we're giving, you know, 1 day of alcohol exposure, but what we're doing is we're inducing a transient ciliopathy. You know, genetic ciliopathies last throughout development, right? You lose that gene, it's not present at any point during development, and so that is a genetic ciliopathy. Here we're giving alcohol for short periods of time, and what I think we're doing is inducing what we have termed a transient ciliopathy. So I'm going to switch gears here, and this will all come together in the end, I hope. And by the way, anybody, you know, if anyone has questions, feel free to raise their hand and ask. I don't mind at all. So a couple of years ago, my lab started looking at cannabinoids, and it's kind of interesting in that, you know, the effects of marijuana on, you know, birth defects has actually been studied quite a bit over you know, the last 50 years or so, and it's been kind of controversial. So there are many studies that have found absolutely no effects of prenatal marijuana exposure. There have been a number of studies going back into the 80s and 90s, and even just a couple of years ago, showing that there are effects in children, you know, sometimes minor effects, school performance, for example, behavior, for example. There are a number of studies going back many years showing that in animals, showing that marijuana does have an effect. What I think is important to understand about a lot of these studies is that since the 80s or 70s and 80s, the potency of THC in marijuana has increased nearly 20-fold. Those, you know, you get a marijuana now, it's oftentimes 20 times more potent. And, you know, that is a testament to natural breeding and selection. And what is also interesting is that, as one would expect in the United States, the use of marijuana has dramatically increased over the last 20 years. So if you look at the use of marijuana, this is in a California cohort of pregnant women. The use of marijuana has gone up quite a bit in the last 20 years. And if you look at these data in a little more detail, it's actually really interesting. If you look at pregnant women over the age of 25, use hasn't gone up that much. It's gone up a little bit, maybe even double, up to 5%. But what's interesting, if you look at women under the age of 24, the use of marijuana over just a couple years ago was about 20%. So 20% of pregnant women in California, this is a California cohort, has increased up to 20% of pregnant women are using marijuana. And so a couple of years ago, we were interested in this, and I had a postdoc who had done her PhD using synthetic cannabinoids. So synthetic cannabinoids were developed many years ago, and they were designed to study the endocannabinoid system. And what's interesting about them is that they're analogs of THC. They're often up 50 to 100 times more potent at the CB1 receptors than is THC. And so they're really good at very selectively activating the CB1 receptor or the CB2 receptor, whichever one you want. And for a while there, luckily this is starting to decrease, but for a while there, lots of people were abusing these synthetic cannabinoids because they were legal, or some of them were legal. And so they were often sold as spice or K2. I'm sure that many of you have seen these. And some of these are still legal. And it's starting to decrease, luckily. But they were really potent, and they were leading to a lot of emergency room visits. You know, I know, you talk to a lot of older cannabinoid researchers. They'll tell you, nobody OD'd on marijuana. It just didn't happen. That has changed recently with the more potent marijuana. But back in the 70s and 80s, there wasn't really a big problem with ODing on marijuana. That was not the case for these synthetics. They were just so potent that people were having psychotic breakdowns. They were having other issues, and for a while there, it was second only to natural cannabis as the most abused illicit substance. And that has changed a little bit. So this is older. I think as we legalize marijuana, I think people are just sticking with marijuana and not using these other things, because a lot of these things come from other places, China, other places, and we just don't know what's in them. Sometimes we don't know how, there are lots of these different synthetic cannabinoids. We don't know which ones are in here. Some are more potent than others. Some have more bad side effects. But we decided to study some of these synthetic cannabinoids. We started with this one. So this CP55940, it is both a CB1 and a CB2 receptor agonist, and it's about 50 times more potent than THC. You compare this to HU210, I'll talk about this one too. It's about 100 times more potent than THC. But we started here with this one, partly because it was one of the better characterized synthetic cannabinoids, and it was also legal. I was able to get it. I didn't have a DEA license at the time. And so we started here, and what we did is we gave this drug within a very acute exposure. So just one exposure on day 8, so the beginning of neurulation. So that period when we don't get those alcohol-induced severe craniofacial effects, but we do get brain effects with alcohol. But we just started on day 8. And then we looked at eye defects as a measure of here. And so here's a whole spectrum of eye defects in the mouse fetus. So we did one injection of the cannabinoid and then looked at face, brain, etc. one of the first things we noticed is that in a lot of our embryos, we were having a lot of craniofacial malformations. So here's a normal mouse fetus with a normal mouse lip and normal eyes and a normal jaw. And you can see that in some of these embryos exposed to this synthetic cannabinoid, we were getting what looked like really FAS-like faces. So we're getting the smooth philtrum right above the lips. We were getting small eyes. In some really severe cases, this looks like that severe mouse fetus that I showed you that was exposed to alcohol. So we were getting almost FAS-like faces in some of our embryos. And we were also getting some holoprotencephaly. So here's a nice normal mouse brain. Here's a mouse brain exposed to a fairly moderate dose of the CP. But you can see quite severe holoprotencephaly. And here's another one. And if you look at the palate, so this is the section right through the face of the mouse. So here's the lip, here's the eyes, here's the palate. If you look in some of these mice, you can see that we have a cleft palate. Here's another cleft palate. There's a really abnormal eye and an abnormal brain. And then we get, here's a cortex. We got some cortical dysplasias, with some higher doses of these CP. And, some of these higher doses are not real. I don't think they're realistic. But what is important is that we get a very nice dose response effect. So just looking at the incidence of abnormal eyes, we get a really nice dose response effect from 2 milligrams per kilogram, which is way too high, to 0.2, which is in 0.125. These are more physiologically relevant dosages of this drug, and that it will cause, psychoactive effects. But even going down to lower doses, and we've even gone down lower than this now since then, and we still see some of these birth defects in the brain and face. And so naturally, the first question that everyone asks us, well, what about other cannabinoids? This is just one synthetic cannabinoid. Is this effect consistent? And so last year we published a study that showed that indeed, if we give HU210, that's the more potent one, that we get effects at even lower dosages. In fact, we had to stop at 0.1 because we were starting to kill embryos higher than this. We obviously wanted to test THC, and we get effects at fairly moderate dosages of THC. So for those interested, a physiological relevant dose of smoking a couple of marijuana cigarettes is right around in here, in between these two. And you'll notice that it goes down. We go up too high, we can actually start to kill off embryos. And interestingly, it's more subtle, but we do get an effect with cannabidiol or CBD oils. I'm sure most people have heard about CBD oils. You can buy them over your, over the counters at your normal convenience stores. And even with this cannabidiol, which supposedly does not have, the regular psychoactive effects of THC, we're still getting birth defects with this CBD. And like I said, it's more subtle, but it is there. So it's the danger is, the possibility of danger is certainly there. Naturally, we're an alcohol lab. That's primarily what we do, is what we're mostly funded to do. So we had to ask, what happens when we administer alcohol and the cannabinoids together? So what we did is we took we in the lab kind of stick with these 2 doses of alcohol. So we have a 2.8 grams per kilogram. That's a kind of a normal high dose alcohol. That's what we usually use to study the mechanisms of what alcohol is doing. But we also will occasionally use a half dose, so it's 1.4 grams per kilogram. And it doesn't really cause all that many eye defects in our minds. It can cause a few long-term behavioral effects, but it doesn't really cause a whole lot of severe malformations. And we took a fairly moderate dose of our CP, the 0.25. And so when we have our alcohol vehicle and combine it with this modest dose of CP, we get a significant amount of eye defects, as you can see here. When we combine it with alcohol, we get this synergistic effects. And so with even a low dose of alcohol, we get this more than additive effects. So even with this moderate dose of our cannabinoid and a modest dose of our alcohol, we get this synergistic effect. And when we combine the cannabinoid with our high dose of alcohol, we actually will affect almost 100% of our fetuses. So it's quite much more than additive in these effects. And when we get the very similar effects with HU210, that more potent synthetic cannabinoid, and we get the same effect with THC. So with THC, we get a certain amount of defects. When we combine it with even that low dose of alcohol, we greatly exacerbate the effects of these two drugs together. And we not only increase the incidence of defect, we increase the severity too. So, we get things like cleft palate and holoprosencephaly, things that we would never see with either of these two drugs alone at these dosages. But we do see when we combine them together, suggesting that there is this synergistic effect. And so we wanted to verify this. And so we turned to our zebrafish colleagues at NC Central, Greg Cole and his lab. I know Ed is quite familiar with Greg. And so they have this story with prenatal alcohol exposure over the years. So zebrafish, as Ed knows, zebrafish are great because they also very nicely phenocopy the effects of alcohol. That they have craniofacial defects, they have eye defects. So here, for example, is a control zebrafish eye. Here's a heavily exposed alcohol zebrafish eye. And over the years, they've shown, they have this whole dose response effect, just like we do in the mice. But we asked them to sort of, see if they get the same effects with this synthetic cannabinoid. And they use rather modest dosages. So first, they kind of looked at just the cannabinoids alone. And here they see that with increasing dosages, they get very similar effects on the eyes that we do in the mice and that they do with alcohol. And so naturally, they took this 0.5% dose of alcohol that doesn't really do much on its own with these lower doses of the cannabinoid and put them together. And they too see the same things. They see a synergistic effect of alcohol on the eyes. And so the great thing about fish is that, sorry, I skipped forward. The great thing about this is that, or the interesting thing here is that, this, just like in the fish, or just like in the mice, fish also have reductions in sonic hedgehog signaling. So we were likely dealing with a very similar mechanism in both the mice and the fish. And so we went back to the mice and asked, well, what are we doing to the sonic hedgehog pathway? And when we exposed to just this acute dose of CP and look, 6 hours later, we get a severe reduction in sonic hedgehog as well as its downstream genes. And this is some very preliminary data, but we get these very similar gene environment interactions with this synthetic cannabinoid, in that we gave a fairly low dose and we get a certain amount of birth defects. But then if we knock out one copy of sonic hedgehog, just like with alcohol, we get this gene environment interaction. And so we were kind of curious as to how this worked, and we went through some papers and looked at, we actually went to Sue Eaton. Sue Eaton had a paper many years ago where this was completely in vitro, but so this is purely in cell lines. But what they did is they gave some endocannabinoids, so the natural cannabinoids in our body, as well as some cannabidiol and THC, and showed that all of these products inhibited smoothing. That's that G protein coupled receptor that sort of is the main driver of the sonic hedgehog pathway once it's activated. A lot of these cannabinoids will inhibit smoothing. So we turned to another colleague, Kevin Williams at NCCU, to see if, because he has some very similar assays, but we wanted to test some of our synthetic compounds and cannabidiol to see if they'll do it. And indeed, all of these compounds will inhibit smoothing in culture. So that is one potential mechanism by which these cannabinoids may be acting, is by inhibiting smoothing and thus shutting down the sonic hedgehog pathway. So going back to fish, one of the interesting things about fish, and a number of people have shown this over the years, is that you can take a single cell zebrafish embryo, inject it with sonic hedgehog mRNA, so it has excessive sonic hedgehog signaling in there, and you can actually protect against the effects of alcohol. So if you inject with sonic hedgehog mRNA and then, a couple of hours later expose it to alcohol, you can almost completely protect against the effects. And so we asked them to see if they could do the same thing with our drugs, so alcohol. And indeed, they do. You see that we give alcohol and the CP together. Without sonic hedgehog mRNA, we get this huge effect. Again, this is looking at eyes. When we inject with sonic hedgehog, we can almost completely rescue those effects. Now, this was kind of interesting in that, these cannabinoids, except for that one brief cell culture paper, these cannabinoids have never really been described to interact with, with the sonic hedgehog pathway. They're really better known for interacting with the CB1 receptors. These are the natural receptors for cannabinoids. And so we asked the natural question of, what if we gave a cannabinoid receptor antagonist? Could we rescue the effects of these cannabinoids in mice? And indeed we can. And when we pre-inject with a CB1 receptor antagonist, we can dose dependently protect against these cannabinoid-induced effects. And our zebrafish colleagues replicated this very nicely in zebrafish, and that they too see the same thing. They can give the CB1 receptor antagonist and very nicely protect against the effects of these drugs. And so that was kind of confused us a little bit, because we could protect against sonic hedgehog by overactivating the sonic hedgehog pathway, and we can also protect by antagonizing the CB1 receptor pathway. And so, these pathways have never really been shown to interact. And so we were kind of curious as to what was happening. So we went in and we kind of pulled down smoothing with, just do a co-immunoprecipitation just to look and see what it was interacting with. And what we did is, naturally, when we pull down smoothing, we get that inhibitory G protein, like I showed early on. That's not a surprise, that we very much get that effect. And alcohol, and to some extent, the cannabinoid can interact with that. But what was also interesting is that we also get a stimulatory G protein. That is, when we pull down with smoothing, it's also binding to it. We can sustain for a stimulatory G protein, which is weird. No one has looked at this much in embryos, but in the adult brain, that has not been shown. But what was really interesting is that when we pull down smoothing, we also can stain positive for the CB1 receptor, suggesting that the CB1 receptor and smoothing are dimerizing in the embryo. And we did some proximity ligation assays and some other assays to verify that. And every assay we've used has shown that in the embryo, smoothing and the CB1 receptor seem to be dimerizing. And so that led us to propose this model. So CB1 and smoothing are both G proteins coupled receptors. And within the primary cilium, they're dimerizing. And CB1 is known to dimerize with a number of other proteins, I think like glutamate and a couple of other G protein coupled receptors. So it's not news that CB1 is dimerizing. It is kind of new that smoothing is dimerizing to things. And what has also been shown in a couple of cases, just a few cases, is that CB1, which normally likes to bind to an inhibitory G protein, sometimes when CB1 forms a dimer, it will undergo a transformation and bind the stimulatory G protein. And of course, these two proteins are going to have contrasting effects on adenylyl cyclase within the primary cilium. And I think under normal circumstances, the CB1 is just sitting here acting as a brake on smoothing. So I remember I showed you early on in that neural tube, you have a gradient of sonic hedgehog expression because sonic hedgehog diffuses. And so, and the various neural cell types in the neural tube are formed based on how much sonic hedgehog they see. And so some cell types in the ventral neural tubes want a lot of sonic hedgehog. Some of them in the lateral neural tube, they want less. And so how much sonic hedgehog they see determines what kind of cell type they are. And so I think under normal circumstances, the CB1 receptor, which we don't really know what it's doing previous to this, is sort of acting as a brake. It's sort of helping to very quietly define and limit sonic hedgehog expression. However, when we give cannabinoids, we're obviously, as us and Sui group showed previously, we're inhibiting smoothing, and we're activating these CB1 receptors that are dimerizing the smoothing. So we're causing this great reduction in sonic hedgehog pathway expression. And on top of that, when we give ethanol is through a process that we're still not quite completely sure of, is inhibiting the function of these primary cilia. And so when we give them together, we're having this triple effect where alcohol is affecting the function of the primary cilia, the cannabinoids are activating CB1 receptor like they're supposed to, and they're inhibiting the smoothing protein. And this is all working to increase sonic hedgehog pathway activity. And so I think that the takeaway things are that, I think our data shows that THC is teratogenic. And I think that as we go through, and more people are starting to do more recent epidemiological studies, which I think is needed because THC is quite a bit more potent and it's used a lot more than it was 40 years ago. And I think CBD too is we need to start looking at CBD. CBD is widely available. It's used, it's been well known that it's used for intractable epilepsy in kids, and it actually works quite well. But it's also occasionally used for morning sickness, which typically occurs around the times of development that we've been talking about. So I think we do need to be careful. But I think we also, I think we've kind of hopefully showed that these cannabinoids and alcohol are converging on this sonic hedgehog pathway through those primary cilia and through smoothing, and through this interaction with smoothing and CB1. And so with that, I will stop. I will thank my lab, Eric and Karen, who did a lot of this work, along with some of the other former members of the lab, as well as some of our collaborators at NC Central. They're great. They all do zebrafish or the cell culture work that we typically do. And so with that, I will, I'm happy to take questions.
Well, thank you very much. That was really interesting in terms of the mechanistic work and also the practical applications having to do with both alcohol and cannabis and CBD. And so it's open for questions now. I had an initial one. So you showed that the CBD receptor antagonist rescued the cannabinoid effect. Does it rescue the ethanol effect?
Yeah, that's a good question. So our zebrafish colleagues show that it does a little bit. We have been unable to do it in the mice because, as you know, with zebrafish, you can very tightly control when you put on the antagonist and take it off. In the mice, if we give alcohol and the antagonist on day 8 at the same time, it actually kills a lot of the embryos.
Oh.
And I think it's because of heart defects. And I think it's just because I think you need CB1 receptor for heart development. And alcohol, we know hits heart development at this time. And you tack on that CB1 receptor antagonist, I think it's just too much. I think it takes out the heart.
Oh, okay.
And it's just because we can't control, in mice, we can't control the timing like you can in the zebrafish.
So does anyone else have a question or a comment? If not, I'll have another question.
I'm sharing.
So you really very nicely showed this gene by environment interaction. Now, what about with people? Are there polymorphisms for the sonic hedgehog or the BAX gene?
For backs, I think it's less clear. I think it's just pretty recently that people are starting to look into backs. With sonic hedgehog, it has been widely it's been widely known that there is allelic variations that can interact with alcohol in kids, and a significant part of my lab is looking for other genes too. Those are just 2 examples. So we're, as with everything, this is multi-allelic. You can have lots of genes interacting, and then you tack on and you might be fine, but then you tack on alcohol, and you cause defects.
Good. Well, thanks. Anyone else? Okay. Now, one announcement I wanted to make is for the registration for the symposium on the 25th, please look in the chat box. There is a listing, or Tyler's put up a link for the registration. So Scott, I really wanted to thank you for your wonderful presentation. Very important work in terms of determining the mechanism of fetal alcohol syndrome and fetal cannabinoid syndrome and their interactions. And anyone who gets in touch, please do.
Yeah, it was always a pleasure. It was good to see you again.
Thank you.
Okay.